I think we can start. Okay. Everyone for coming. Good morning. Today we have Maria Cortés. She's from the Marine Biomolecules group. So, Maria, thanks for coming, and the floor is yours.
Thank you. Hi. Thank you for the introduction, and thank you all of you for coming to the talk. I hope you find it interesting. I'm gonna. Uy, un momento, me oigo a mí misma. Vale, yeah. I'm gonna talk about processes involved in organic carbon cycling and transport, and the relationship of those with microbial interaction in marine environment. And just as a brief introduction on my scientific career, am I covering the screen? As a brief introduction on my scientific career, I did a bachelor in chemistry in the University of Barcelona. And then I moved to London to do a master in supramolecular chemistry. And after that, I started my PhD at IMEDEA. And then I did a number of postdocs outside, one of them in another institute at the CSIC, in the, can you see the mouse? Yeah. In the Instituto Andaluz de Ciencias de la Tierra in Granada. And the rest of them were stays in international centers, in 3 of them, in the University of California, in the King Abdullah University of Science and Technology in Saudi Arabia, and the Max Planck Institute for Chemistry. After spending 4 years in the Max Planck, I finally returned home. I am a native from Mallorca, actually. And with Maria Zambrano contract at the University of the Balearic Islands. And then I got a position as a científico titular in the CSIC, and I started here at IMEDEA a little bit less than a year ago. So when I first started my scientific career in London, I was doing a master in pure chemistry. It had nothing to do with marine science, and we were basically synthesizing a very specific set of organic molecules in the laboratory, and we were then trying to crystallize those molecules and look at the interactions in between molecules. So we were focused on low energy interactions such as hydrogen bonds, but those interactions were the ones that were holding the supramolecular structure in a very particular way in the 3-dimensional space. So just so you know the background that I had before I started in marine science, I was working with complex organic structures, and then I started working at IMEDEA. I started as a technician, and I was measuring the concentration of inorganic nutrients in seawater. So I was measuring nitrate, nitrite, and phosphate at the time. We were doing it by hand because we didn't have the flow injection analyzer. So there was a small number of samples that we could do every day. But so I was coming from complex organic structures to measure small inorganic molecules in the water. However, these very simple organic molecules are fueling very important processes, such as the fixation of carbon dioxide from the atmosphere in the surface ocean. And here you have this apparently very simple equation, but it holds a lot of complexity inside, which is how phytoplanktonic organisms are fixing carbon dioxide and converting it into organic carbon in the surface ocean. And at the same time, there is constant respiration of heterotrophic organisms. And when I started my PhD, so I went from technician to PhD student, I focused on how this small inorganic carbon dioxide molecule that was coming from the atmosphere was converting was being converted into organic matter, and which is a step where we are going from a very simple molecule to very complex organic matrices, where there are a number of very different organic compounds within what we are naming here the average organic matter isomer. So I soon understood that in order to study CO2 fluxes between the ocean and the atmosphere, and also how all this carbon is being cycled, we had to pay attention to the microbial diversity and the microbial functioning of the surface ocean. So here we have in big numbers how much pentagrams of carbon is being fixed in the surface ocean by phytoplankton organisms, which is 50 pentagrams of carbon per year, which is half of all the fixed atmospheric CO2. The other half is done by land plants. And half of what is fixed is being recycled in the surface ocean, and the other half is being transported vertically to deeper waters where it remains for longer periods of time. And if we want to understand how all these processes are occurring, we need to study not only the cycling that is occurring in the surface ocean, but also processes that are involved in the transport, the vertical transport of this carbon. So here in this talk, I just selected out of all the different studies I've been doing, I selected a few of them. One is focused on air CO2 fluxes, which is coming from my PhD and then returning to that during my time in the Max Planck Institute. And another one is related to DOC dynamics in the epipelagic and mesopelagic layers. So can I? If we look at processes that are regulating air CO2 fluxes, we have to consider, of course, changes. This is the formula that is used to calculate air CO2 fluxes. And of course, we have to consider the changes in water temperature and salinity, which are determining S, which is the solubility of CO2 in seawater. And we have to also consider the gas exchange coefficient, or the velocity at which this gas transfer is happening, which depends a lot on wind speed. We basically parameterize this factor as a function of wind speed. Although we know that there are many other processes affecting it, such as the accumulation of surfactants or hydrophobic molecules in the microlayer of the ocean, which is suppressing this gas exchange because it's decreasing its velocity. But what I'm going to focus on now is how the metabolism of the community inhabiting the surface water was affecting the CO2 PCO2 gradient. So these terms here represent the thermodynamic term of the equation, and it's basically the difference between the PCO2 in the water and the PCO2 in the air. So we know that changes in PCO2 in the air are much slower than changes occurring in the water. So this gradient is basically controlled by changes in the water. And these changes in the water depend on physicochemical factors, such as changes in temperature, but also on the biological activity. So in a number of cruises that we performed in the subtropical North Atlantic and also in Antarctica, so in very contrasting ecosystems, we were measuring the gradients, the PCO2 gradients, and we were also looking at the net community production by using Winkler incubation bottles, day and dark incubations at in situ experiments, and we wanted to check whether there was a difference in controlling this PCO2 in the water between the microhabitat or microorganisms inhabiting the top surface of the ocean. So we were sampling in the top 2 centimeters compared to the ones that were inhabiting the 5 meters of the ocean, which is what we normally use as the surface ocean. So what we found, there is way more work behind it, but I just wanted to summarize it with this graph. This is we have represented the delta PCO2, so differences between water and air, and the net community production in the top centimeter. So first, we found that the metabolic rates, so respiration rates and production rates in the top centimeters were higher than the rates that we could find at 5 meters depth, and that the PCO2 gradients were controlled by this community, not the one at 5 meters depth. And even though this may look like very few points in this graph, each of these points involves very challenging logistics. We were in open seas, and we had to deploy a Zodiac to do the PCO2 measurements in order to be away from the big boat fumes contamination. And we were also incubating the Winkler bottles in situ by deploying a drifting buoy that was recovered after 24 hours. Some of the bottles were in dark and some of them in clear transparent bottles. So what we can see here in the graph is that positive numbers are resembling when production is higher than respiration, and this is where we have the negative gradient. So a negative PCO2 gradient means that the ocean is acting as a CO2 sink. So we could see how, as we would expect it, the net autotrophy is when we have net autotrophy, the ocean was performing as a CO2 sink. And we could also see how when we had net heterotrophy, which is represented in red, when respiration was exceeding production, we could find one of the points where the ocean was acting as a moderate CO2 source to the atmosphere. And this was published when right after I defended my PhD. And the work inspired some research that I did after 12 years in the Max Planck Institute, where I was working in a sailing research vessel that the Max Planck had custom made. This is a platform of about 22 meters long, where 50% of it is used for laboratories. We had Teflon tube inside the keel to pump water from 2 to 3 meters deep, and what we call the snorkel, which is this, which was pumping gases and bringing them to the atmospheric lab. So we have an ocean lab and an atmospheric lab, and we were recording with a big number of different sensors, a lot of geochemical parameters, as well as biological data. So we were also able to sample water down to 1,000 meters. We had an A-frame built here and a rosette sampling and everything. But what I want to focus now is that what I want to pay attention to is that we could measure at much higher resolution than before we could get the PCO2 gradients in the surface water in the low atmosphere in continuous. And we also have a minimass spectrometer on board called mini-Ruby, from which, through membrane inlet mass spectrometry, we were able to measure argon, dissolved oxygen, and dissolved nitrogen. And the thing is that when you measure argon, because it has very similar physicochemical characteristics than dissolved oxygen in seawater, the solubility of it is very similar to the solubility of oxygen. And then this allowed us to have the total oxygen changes, the total oxygen variability measured along the transects that we were sailing, together with the argon variability. And when subtracting the argon to the oxygen, we can estimate the oxygen supersaturation in the surface, which is directly related to the biological activity. So with these two different kind of measurements, we could get high resolution data on PCO2 gradients and PCO2 fluxes, and high resolution data on net community production. This is where we were sampling. We had a long latitudinal transect along the northeast Atlantic Ocean. We were in yellow color. You can see some stations where we did some deeper analysis of the whole water column, and in blue you can see the transect. So we were sampling a very big gradient of different very cold and highly productive waters near the North Pole, all the way to very oligotrophic and warm waters near the equator. So this is the data that we could get out of the high resolution measurements. Here in the left, we have the fugacity of CO2 in the water, in the air, and for comparison, I plotted the delta oxygen to argon ratio. And not going to pay too much attention to that, but here you can, I don't know if you see the numbers, but the variability of PCO2 in the water goes from 250 to 450, whereas the variability in the air goes from 385 to 415. So again, as we would expect, the variability, the changes in the air are much smaller than changes in the water. And again, PCO2 gradients are driven by changes in the water. And this is part of the data that we got, where you can see represented in the x-axis is latitude. So from the right side to the left, we go from high latitudes to lower latitudes. I'm thinking this way because this is in time how we did it. We went north to south. And as you can see, we were in the top panel, we have chlorophyll. So we were in very high productive waters in the northern part of the transect. And then we crossed the front at around 45 degrees north here, where we went from moderately productive waters to very oligotrophic waters. And here around 20 degrees north, we were crossing the Mauritanian upwelling, just to give some context. And here I have represented the calculated CO2 fluxes that we could estimate out of the data that we got. And here I have represented the net community production that we could estimate it from the mass spectrometer data. And this looks like very simple, but this is actually unprecedented high resolution data that we could get. And you can see how the fluxes and the net community production are mirroring each other. And you have to think that these two are estimated from independent measurements. So that was really nice to see. So we reproduce the same kind of diagram, the same kind of figure that we did in 2007 with this much better data set, and this is what we found. So the tendencies are the same ones, and we could again we could see much better how most of the transect that we sailed was, the ocean was acting as a CO2 sink, and we had net autotrophy. And but there was a little fraction of it, and this is mostly in oligotrophic ocean. So the colors here represent chlorophyll A in logarithmic values. So bluish are samples taken from very oligotrophic waters, and reddish are samples taken from much productive waters. So in the oligotrophic waters, we could see some net heterotrophy and moderate. The ocean was acting as a moderate source of CO2 to the atmosphere. And we are still working on the data, so we think that the deviations from the model are mainly probably, or we expect they are linked to different microbial composition. So different, the diversity of the both autotrophs and heterotrophs in the waters that we were sampling, and we are working into this data now. So I've been talking a little bit on, I don't know the time, but on this work that we did with RC CO2 fluxes. For me, it was very nice to go from this very low resolution data set to this very high resolution data set and see and extract more information out of the same theoretical context. And now I'm going to talk a little bit more about DOC dynamics in the water column. So I'm going to change subjects a little bit. So I want you to think about, once this atmospheric CO2 is trapped by the surface ocean, it's, as I mentioned before, it transforms into organic carbon. And it can be in the form of marine biomass, of particulate organic carbon, or of dissolved organic carbon. And here, when I talk about particulate or dissolved organic carbon, I mean the abiotic part of this carbon. So of course, the marine biomass can also be part of the particulate, but when when I talk about marine biomass and particulate organic carbon, I mean the biotic fraction of it in the marine biomass and the abiotic fraction of it in the particulate organic carbon. And well, in a drop of seawater, we have 10 to the 6 microbes or bacteria, and more than 100,000 different dissolved organic molecules. And if we have a look at the organic carbon inventory in the oceans, we can see how DOC is mainly most of it. And this is why I focus my research on DOC. So most of the organic carbon hold in the ocean is in the dissolved fraction, and most of it is in waters deeper than 200 meters. If we compare it to the magnitude of carbon that we have in the atmosphere as CO2, we can see that the magnitude of this CO2 or the magnitude of the DOC is very similar to the magnitude of the atmospheric CO2. So small perturbations in resident time of this DOC could have big consequences for atmospheric CO2. And we have to think that DOC is not only the biggest reservoir of organic carbon in the ocean, it's also the biggest bioreactive reservoir of organic carbon in the ocean. So it's in constant interaction between the microbes that inhabit the waters not only the microbes, but also big organisms can be excreting or exudating organic material to the media. I'm focusing mainly on the microbes because they are the most important in the open sea. And there are also constant change between dissolved and particulate material because of sometimes aggregates form and dissolve goes into particulate phase, or sometimes particulate material can dissolve can be dissolved into the dissolved phase. So it's hard to understand all these processes, but it's important to look into them. So yeah, this is mainly why I will focus now in the ocean dynamics, because the ocean is the largest reservoir of organic carbon in the ocean. Small changes in resident time could cause disturbances in atmospheric CO2, and this is also the most complex pool of carbon in the ocean and is the least known. And the reason why it's the least known is because there is this mismatch between the inventory, so how much carbon do we have and in which form do we have it, and fluxes. So here in the right pie, you can see how most of the inventory of organic carbon is in the form of a refractory pool. So it stays in the ocean for very long periods of times. And the small fraction of labile carbon is the one that is responsible for most of the fluxes. And if we want to understand this cycling and the transport of this carbon into the deep sea, we need to we need to do a multidisciplinary work where we look at chemical composition of this material, at the interactions with the microbial community, and also at physical processes that are telling us more about lateral and vertical transport. So when I moved when I started working at the Red Sea, when I moved to the Arabic Peninsula, I realized, that was 2015, that this is a map where you can see modeled DOC distribution in the Indian Ocean. So this is Africa, this is the Red Sea, and yeah, the Indian Ocean, the Persian Gulf, etc. And what you can see is that this was published in 2009, but still when we arrived there, there was virtually no DOC data published in the Red Sea. So we decided to start getting some data there. And we established a station offshore in the central Red Sea. This was a station of about 700 meters there. So we were covering epipelagic and mesopelagic waters. And the Red Sea, it's considered as a model for the future ocean. It's like an ocean in miniature. It has a gradient in oligotrophy. And it also has upwelling events in the south, and it holds quite a lot of heat. So temperature and salinity it's higher than in other oligotrophic oceans, and the concentration of oxygen is smaller than in other oligotrophic oceans. So we decided to start working on that station, and we were going there to sample every month, getting vertical casts every month from the whole water column, which was from the surface down to 700 meters for over 2 years and a half. And the first thing that we did was to create an inventory of how much DOC did we have in these waters. So this is the vertical distribution of DOC in the red panel. the different colors represent different seasons. And we found concentrations of 70 micromoles of carbon per liter averaged in the epipelagic, and concentrations of about 51 micromoles of carbon per liter in the mesopelagic, which was very reasonable when comparing to other oligotrophic waters. What was intriguing was the high variability that we found in deep waters that we were not expecting. So you would expect more variability because of all the cycling in the epipelagic, but then narrower variability in deeper waters. So just to keep that in mind. We could check on slight seasonality that we observed in the epipelagic waters, where there was, which is what you can also observe in open water stations such as baths or hots, where there was an imbalance between production and consumption during the summer of about 8 to 10 micromolar, which is the carbon that can be potentially, that is not being recycled and that can be potentially transported to deeper waters. Part of this carbon is semi-labile and will contribute to respiration in deeper waters, and the rest of it would be refractory and it would just accumulate in the deep waters. So I'm not gonna go too much into detail because of the time, but we could have these maps of changes in dissolved organic carbon with time. This is time. And you can see the cast that we were sampling. You can also see how we could have higher resolution data, of course, because we had the sensor of oxygen from which we calculated the apparent oxygen utilization. And from DOC and apparent oxygen utilization, we investigated the relationship between these two using different isopycnal layers. And of course, in the surface, we didn't find any good relationship between these two, because in the surface you have both oxygen consumption, oxygen production, plus the oxygen exchange with the atmosphere. But we found this isopycnal layer, which was between 100 and 200 meters, where there was a significant relationship between the DOC consumption and apparent oxygen utilization. And we could calculate that the microbial remineralization of semi-labile DOC within this isopycnal contributed to 20% of the dissolved oxygen variability in the water column. Which is, so you have to think that when there is remineralization, the vertical transport is being attenuated. So the potential of carbon for being transported is attenuated by microbial respiration. And we were also doing not only this monthly cast, but we were doing dial cycles during the different seasons, and we were doing 2-hour casts every 24 hours in that same station. And we found unexpected changes between day and night. So this is average data of daytime and nighttime. Daytime is represented by the empty circles and nighttime by the solid circles. Here you have heterotrophic bacterial abundance, cells per milliliter. Here you have the DOC concentration. So these two bulk parameters were not showing significant differences between day and night. But when looking at more specific parameters, such as the percentage of high nucleic acid bacteria and the spectral slope of homophoric dissolved organic matter. Sorry, because I didn't introduce these terms, but this is a way to, the spectral slope is a way to look at differences in the organic matter size. So smaller slopes represent higher molecular weight DOC, and bigger slopes represent lower molecular weight DOC. But I don't want to go too much into detail on that, but I wanted to notice that both in the percentage of high nucleic acid bacteria and the slope ratios of sedum were significantly different, the day data from the night data in the mesopelagic layer, which was again very intriguing. Then we had a look at, so this is day and night data average, and here we have epipelagic and mesopelagic data average. And this is dial cycle, which are not, the error bars are quite deep, but these are tendencies that are very hard to see, especially in oligotrophic waters. And but we could identify dial cycles both in the epipelagic and in the mesopelagic, as you can see here. You can see here the changes of the percentage of high nucleic acid bacteria and the DOC concentrations in both layers. And we published some data, some articles with this data. And because we were intrigued about what could be happening that was generating this high variability of DOC in mesopelagic, and also the day and night differences in the microbial, in the bacterial community and the sedum spectral slope, we realized there were some colleagues of us that were working with acoustic data, and they had an echo sounder moored in the bottom of the same station that we didn't think of. And when they looked at the data and they shared the data with us, we realized that we had in that station a very, very intense deep scattering layer. So this is an echogram. For those who are not familiar with it, is representing the movement of dial vertical migrating animals. So this is nighttime, and the middle of daytime. So what you can see here is this deep scattering layer that is giving us information on that these migrating animals were staying in between 400 and 600 meters during the daytime. So basically, this is telling you we think that these animals are migrating every day. They are feeding during the nighttime in the surface. Probably there are different theories that I really don't know which one is the valid one, but we believe it's because they are escaping from predators. So they go up to the surface during the night, they feed at night, and they swim very fast. So in about 20 minutes, they are from 200 meters down to 600 meters. They swim very fast during when the sun goes up. Stay in the deep waters during the whole day and swim very fast again at night when the sun sets to stay and feed in the surface waters. And this is something that is happening in all ocean basins. So here you have echograms taken from the North Atlantic, Indian Ocean, West and East Pacific. And these are just like snapshots occurring during 24 hours. And what you can see here is that in some of the basins, not all the animals are migrating. So here you can see how some of them are remaining during the nighttime in the deep scattering layer. But for example, here in the East Pacific, all of them are moving up and down, and none of them are remaining during the night in the deep waters. And this is actually what we saw was happening in the Red Sea. This was an echogram that we saw over and over. It's not just that one day. And what is interesting is that it's really generating a very big difference between day and night in deep waters. Okay, so we asked ourselves if these diavertical migrations in the Red Sea could be transporting bioavailable or labile carbon from surface water to the mesopelagic waters, and if this could be altering the DOC composition and microbial activity down there. The migrations in the Red Sea are mostly from Lenthosema pterotum, so it's mostly fish in that case, but in other oceans could be also a lot of zooplankton involved in the migrations. So in order to answer this question, we decided to isolate water from the surface and isolate water from the deep scattering layer, 550 meters, I think we sampled, during the daytime. We did a number of experiments. This is one of them. And what we did was to isolate this water and to have experiments where we were looking at how the heterotrophic bacteria or heterotrophic prokaryotes, because we have to consider they were also archaea there, were making use of the dissolved organic carbon in the water. So we were incubating in the lab the water that we took from both layers, and we were looking at how fast the DOC was being consumed and how fast the bacteria was growing. So how fast and how big the bacteria was growing. And we could see that actually in the deep scattering, the water incubated from the deep scattering layer was generating bacteria that were bigger in biomass. So this is bacterial biovolume here, which is directly related to the bacterial size. And we could see how this is the start of the experiment and the end of the experiment. The incubation lasted about 6 days. And we could see how bacteria were growing bigger in the water from the deep scattering layer than in the surface water. And that was generating growth efficiencies also higher in the deep scattering layer than in the surface water. So we published this paper saying that probably these diavertical migrations are generating a hotspot for heterotrophic prokaryotes in the Red Sea mesopelagic water. So what we think is happening is that because they are consuming feeding during the night in the surface, once they go down here, they literally stay there and they metabolize what they have been eating. So just by being there, they are exudating organic matter to the media, but also they could be defecating what have been metabolized out of the ingestion of material from the surface water. And we believe that this within this action, they are transporting a small portion of labile dissolved organic carbon that is feeding the heterotrophic bacteria within this layer. So they are moving this small part of this small fraction of labile carbon, which is generating a flux within this deep scattering layer and generating a hotspot of bacterial activity. Now, this piece of work inspired new projects. For example, this is the Shelida project that I'm involved with. It's a Plan Nacional where we will try to, we will look at different vertical transport processes such as gravitational, migrational, and physical transport. And this is going to be done in the Bay of Biscay in the context of the RATCAN cruises. So this is in collaboration with the Instituto Español de Oceanografía in Gijón, in the Centro Oceanográfico de Gijón. And we will have the opportunity to sample in an offshore station, which covers down to 4,000 meters. So we will have the opportunity to look into how diavertical migrations occurring there could be affecting also this ocean basin, and whether this process that we observe in the Red Sea could be also happening here. We will cover not only epipelagic and mesopelagic waters, but also bathypelagic waters. And we will see, because one particularity of the Red Sea is that the deep waters never go lower than 20 degrees Celsius. So of course, temperature has an effect on the processes that we observe. And here the temperature goes down, I believe, to near 10 or 12 degrees Celsius, which will make a difference. I also want to finish saying that, of course, the DOC is involved in almost all biological processes in the open ocean, but also in the coastal system, it has it's also important because it's in the context of blue carbon ecosystems, such as seagrasses or mangroves or coral reefs. The coastal system has the capacity to bury carbon in the sediments and keep it there for very long time periods, such as the deep waters in the ocean, as far as these systems are not altered. And they are also the source of carbon export to the open sea. And the organic carbon hold, or the dissolved organic carbon hold in the sediment, is also feeding different types of microbial metabolism that could be generating subproducts such as methane or nitrogen oxide or CO2 that could be contributing to greenhouse gases, and also contributing to the production of different other toxic gases such as hydrogen sulfide, which could be detrimental for the ecosystem. And I'm also I'm collaborating on the SEGMA project, which is a project from Julia, funded by CAIP, and I just got funded myself a project to work on Solenio project, I called it, to work on Cabrera in the concept of the Parques Nacionales funding. And I think, just to summarize, that we have to focus on both observations and monitoring in order to understand carbon cycles in specific ecosystems and also in the global ocean. And it's important to do so at different time, spatial and vertical scales. And I want to point out the power of high resolution data for that. But we also need to work in another level, in an experimental level, if we want to understand the mechanisms behind these observations to better estimate these carbon fluxes. And in that sense, I think it's important to have a closer look on the chemical composition of the DOC, which is a big gap that we are always facing. I have reported here some data related to CDOM and FDOM, which are the optical characteristics of dissolved organic carbon. I have also worked with the quantification of amino acids within the dissolved organic carbon material as representatives of labile molecules. But there's still a lot of work to do if we want to understand the chemical diversity of these molecules and how are they interacting with the microbial community? And I hope that in that sense, we can do a good job here in the metabolomics service that we are trying to set up, which has a lot of potential for that. And thank you.
A couple of questions. Thanks. It was really very interesting. So I think the questions make sense. Okay. So one was on this transport of organic carbon by zooplankton and fish, basically, which is very interesting. And I was wondering whether, just my ignorance, at that depth, right, the respiration of the bacteria, is that limited by availability of oxygen? And could it happen that under certain circumstances, this extra input of carbon leads to, say, hypoxic conditions?
It could. We never got into a hypoxic condition, so because we measured here the dissolved oxygen. Well, this is not dissolved oxygen straight, but the apparent oxygen utilization. So we know we didn't have hypoxic conditions, but it is true that in the Red Sea, contrary to other in other ocean basins, instead of having, you normally have this oxygen minimum zone, and then oxygen increases again with depth. Generally, okay. What happens in the Red Sea is that you get this oxygen minimum zone, never hypoxic, but minimum but low oxygen levels, and then they stay low for the whole water column. So it could be that this transport of carbon, together with the temperatures that we have down there, which are quite high compared to other deep water temperatures, could be generating this low oxygen concentrations related to what you just mentioned. But it's hard to, it's hard to understand all the mechanisms behind that.
Another question.
There are actually a lot of questions about why we have this oxygen minimum zone in general. So.
No, please go ahead.
So now we know that some pelagic fishes are changing their movement patterns because of the climate change and temperature, other things. So I'm guessing that maybe you have questioned how this change in these patterns can affect the dog or semi-labial availability, and it maybe can fix the synchronicity with other organisms that use the dog. And I don't know. It's just a question that maybe you have had that question.
You mean how the changes that are involved in a warming scenario could affect?
They are affecting. We know that they are affecting these patterns of migration in the depth of these pelagic fishes. So maybe if you, since I understood that your conclusion is that these organisms that move to the deeper and exudates and all that components are used for other organisms that use this as in the dog cycle, that maybe this can affect, and I don't know.
Yeah, definitely. I think they can. The problem is that things are moving faster than we can understand. So these are actually very basic questions we are trying to answer with these observations, and but the climate change is occurring. So I believe so. I believe they are affecting, but it's hard to have a global picture on how this is affecting globally. We should look at these processes in all the oceans in order and have here you have so this is, I think, also this is high resolution data on acoustic high resolution data here. So I think this is also very important. If we could have this data from different ocean basins to know how this is changing over time, that could help answer this question. But it's not that easy. To have this kind of data, you need to have a moored echo sounder, and you cannot have, I don't know how deep they can go, or the deeper the ocean, the harder it is to have that. Normally, the echo sounders are in the bottom of the ships, and you just monitor the data while you are moving, and then you reconstruct. And if you want to know more about one system, you stay 24 hours. For example, here, these echograms here were taken during, I believe this was taken during the Malaspina project, where they were just staying 24 hours in one station, and they could gather this information out of the echo sound from the top of the boat. So, yeah. It would be great to have the same way we have satellite data that allow us to see general patterns in the surface ocean. It's very hard to have this kind of high resolution data from the bottom of the ocean. But I think the scientific community is moving into that, in the sense that we have Argo floats all over now, and which can help us reconstruct some biogeochemical parameters. And yeah, acoustic data is still limited though.
Thank you for the presentation, very nice. I would like to know if you have an idea about the general numbers in this global number of the change of carbon, the upper layer, the role of the sub-scale or mesoscale processes, physical processes, like the one that you were showing, one figure that we found in the Algon Sea, is abduction. So how important is this mechanism, or do you have a number of export of the upper layer? This is from the deep chlorophyll maximum of 50 meters and subduction until almost 150 meters. So do you have an estimation, or do you know any paper on that?
I personally don't know, but I think this is a very big gap that we still have to fill. So we are identifying new processes that are involved in vertical transport of material from the surface to the deep ocean. For many decades, we have focused on the gravitational flux, so the sink of particles or refractory material that is just vertically sinking to the ocean. And we are now identifying other processes that are affecting this vertical transport. And this is one of them, right? The submesoscale transfer related to eddies, right? And we also know that eddies are, well, you know better than me, but the frequency of eddies is increasing also. And so, no, this is something really, really interesting that we should be looking into. It's also hard to model how important these eddies are and how much carbon are they transporting. But this is also an example of high resolution data that could be very, very beneficial to explore in order to estimate how much they are affecting vertical transport of material from surface to deeper waters. Yeah, especially because at the depths that these eddies can get, material from the surface to the deep water, there is still a lot of microbial activity. So they are both transferring carbon to deeper waters, and they are probably also feeding the bacteria down there, because this rapid subduction of chlorophyll, this is fresh material, this is labile material. So I think it would be really interesting to work on both, how often these processes are occurring and how are they affecting not only the vertical carbon transport, but also the potential remineralization of this carbon in these deeper layers.
Yeah. I have another question. Unless anybody.
I have a question.
Okay. No, go ahead.
We have
I
time.
didn't see your hand. Sorry.
Yeah, thank you. That was very interesting. I would go back to the first part of the talk and mix it with the second one.
Okay.
So I was wondering if what is the role of organic matter in this flux of CO2? Because I mean, my understanding is that in many cases it can accumulate and make these surface leaks, and this can change, the roughness of the boundary layer, but also maybe gas exchanges. So did you look at it, or do you suspect that there is some relationship between primary production and the efficiency of this process?
Yes. Do you mean like accumulation of organic material in the surface?
Very certain.
I have that. No. It's no. I think I thought I could talk forever, and I had to remove a lot of here. So one of the here. Yeah. Actually, I was looking into. I don't know if it's exactly what you asked, but when we were taking water from the very top centimeters of the water of the surface and comparing it to water at 5 meters, we were also taking the organic carbon. We did not limit the measurements to dissolved. We took the total organic carbon because we wanted to see exactly that, how much of the total organic carbon could be affecting the gas exchange. And what we saw was an enrichment in the top centimeters related to 5 meters. So there was more material in the top centimeters. And this represents the gas exchange coefficient. Well, so the higher the concentration of organic material in the surface, the lower the gas transfer velocity. So when you find...
So, but is this normalized somehow by phytoplankton biomass, or because you would expect both things?
Of course. No, we did not. No, we did not normalize it. Yep. So we didn't. This is total organic carbon. So we did not separate here the microbial biomass versus the abiotic carbon. But what we did was to look at the net community production, which I just showed, to see whether to see the effect on the CO2 fluxes. So what we basically saw is that when you have an accumulation of organic material in the very surface, this is having an effect on the kinetics of CO2 fluxes. And when you look at the net community production of the microbial community, this is having an effect on the PCO2 gradients, which are two different factors of the equation. But the processes are interrelated. It's hard to separate one to the other. I think because this was done with in situ measurements, I think that in order to answer your question, we should run some experiments in the lab, controlled experiments in the lab, just with abiotic material, and then mix it and see what happens, mix it with abiotic plus biomass and see what happens. But yeah, we had the but in any case, there are other studies that have looked into the accumulation of particularly surfactants, so hydrophobic molecules in the very surface of the ocean, which can be very important when you have an event of oil spill, for example, or even when you have a lot of atmospheric dust that is being deposited in the surface ocean. So these are events where this suppressing effect could be more important. Yeah. But at the same time, it's not only organic material, abiotic organic material that has been accumulated. Also, when you have dust events, you have airborne bacteria that are being transferred and doing their thing in the surface. So yeah. Yeah, more experiments.
Now that you say about the cultivating, the culturing the bacteria, when you showed the graph about the size of the bacteria, you said that you took one from the flat layer and another one from the surface, if I am correct.
Yeah.
How did you culture them? Or like, do you mean you isolated the bacteria and then, or you took just the whole, because I don't know how you measured it, just took like the water and keep it like that, or you sort it and then isolate it?
We took water from both layers. First, we filtered it through 0.2, so we had only dissolved organic matter there. And then we took another more volume of water and we filtered it through 0.8. So we would have the dissolved organic carbon plus the heterotrophic bacteria there, but we were removing any potential predators to avoid the effect of predators in our experiments. And what we did was we used this isolated bacteria, so 0.8 filtered water, we inoculated into the 0.2 water. Normally in a 1 to 10 volume ratio. So we have, we increase the amount of available carbon per cell in the experiments in order to see a change, because otherwise it would be very, very difficult.
And you culture that for how long?
We culture that for about 6 days.
Because in my understanding, there is a lot of bacteria that you can grow. So do you think that the numbers you have there, I mean, is there a way of knowing that what you were actually growing is what actually was there? I don't know if I...
Yeah, I understand. Yeah, we have the microbial diversity data. I can show you, I can't remember. But yes, we had the initial culture or the initial diversity of bacteria, and then some of them grow better than others. So they're probably, because, you know, once you take the water and you put it in a bottle, you also have the bottle effect. So this, I know for culture, I don't know exactly what you're doing, but it's very different to culture organisms in the lab than using environmental samples. But yes, we do observe this bottle effect once you take the water out of the, once you put the water in a bottle and then see how it's growing. But this is the best that we can do, and for that you have the controls and yeah, the comparison between the two layers is still valid. Yeah, this is how people do it also. Yeah. But I can explain you better or send you the paper if you want. Yeah, no, we were extracting DNA and looking at 16S. sequencing, but I didn't show the results here.
Last question?
Oh, thanks. So, again, this might be completely stupid question. So going back to things that also Oscar was touching on before, the top layer and the exchange of CO2. So again, I'm not a chemist. Okay, CO2 in water becomes bicarbonate. There's an equilibrium, right?
Yeah.
And this equilibrium depends on the pH.
Mm-hmm.
But you never talk about pH in, is that important at all? Or is that a factor that we should not think about? But for example, changes in temperature are going to change pH. And I don't know whether this then, the availability of CO2 versus, for example, bicarbonate is going to change the efficiency of photosynthesis of the photosynthetic cells there, which therefore will impact on the exchange of CO2. Is that something we should think about?
I think this is something we should think about in the long term, because the small changes in pH that we could
Exactly.
If we look at CO2 data in the water from 20 years ago and from 20 years from now, we could see changes in pH that affect to these small changes in the CO2. But not within the time scale that we were working. Yeah, it would be way more, way bigger the effect of biological activity than even though changes in pH are also affecting the biological activity, and it can be detrimental to certain organisms more than others, but not in the time scale that we were working. Yeah, because the measurements are, yeah, it's flux measurements are up to 30 minutes.
Great. So thanks again, Maria. Thanks